ఏంటి మసాలాస్ మినిమైజ్ दिसणार ना नाही मला हे वाचायचं आहे ना मला इथूनच सुरुवात करायची आहे ना है ना दिसते मी पोझिशन बरोबर आहे
Yes. Hello all participants. This is Dr. Vijay Nilkan from Vetina Healthcare. I welcome you all in the first webinar in association with the Maharashtra Fishery and Animal Science University Nagpur and Nagpur Veterinary College. I welcome you all the participant and I am hand over this all control to Dr. Shubhangi ma'am. She will introduce to guest and she will conduct the next sessions with you. Ma'am, over to you.
Yeah. Good morning, dear Dr. Sadanand Fuljale. Good evening, friends from India, Nepal, Bangladesh, Argentina, and good morning, friends from USA. I welcome all the participants to this international webinar organized by Department of Veterinary Microbiology and Animal Biotechnology Teaching and Research Cell, Nagpur Veterinary College, Nagpur, which is a constituent college of Maharashtra Animal and Fishery Science University, Maharashtra, India. It is my great privilege to introduce my friend, Dr. Sadanand Fuljale, Assistant Professor from USA. Dr. Sadanand native is in Maharashtra, India, received his DVM degree from Nagpur Veterinary College, Nagpur, Maharashtra in the year 1997. He completed his master and PhD from Indian Veterinary Research Institute, Izzatnagar, UP, India in the year 2001 and 2004. He has been awarded with ICAR NET Award by Indian Council Agriculture Research, New Delhi, IBRI Senior Research Fellowship during PhD, IBRI Junior Research Fellowship during entire duration of MVSc, and Merit Fellowship for Academic Excellence during entire duration of DVM. Dr. Sadanand has implemented several research projects. He has completed 2 projects as a principal investigator and 1 project as a co-investigator. Presently working on 5 projects as a principal investigator and co-investigator. Dr. Sadanand has member of master's and graduate students thesis, mentor and in committee of 29 students and 6 trainees, work as a research assistant, research associate and postdoctorate fellow under his guidance. Dr. Sadanand has on editorial board of Stem Cell Research, Development and Therapy, Stem Cell International, Journal of Stem Cell Research and Transplantation, Biomedical Research International and World Journal of Publisher, also reviewer of many journals. Dr. Sadanand published 74 research papers. His H index is 24. His main area of research is diagnosis and therapy for osteoarthritis, stem cells and microRNA. He is multi-talented researcher and academician. Dr. Sadanand also worked as an assistant research scientist in Augusta, USA during 2009 to 2012 and assistant professor in the Department of Orthopedics in Augusta, USA during 2012 to 2019. Dr. Sadanand presently working as an assistant professor in the Department of Medicine, Center for Health Aging, Cell Biology and Anatomy, Medical College of Georgia, Augusta University, USA. As per Indian tradition, we wholeheartedly welcome Dr. Sadanand Fuljale. It is our immense pleasure to have you here, dear sir. Today, Dr. Sadanand Fuljale deliver webinar on COVID-19 virulence in each patient might be impacted by the host cellular microRNAs. I am sure this will be a great intellectual engagement. It will be my humble request to all the participants to enjoy the presentation. You may put your questions through the chat windows. All the best. Now I hand over the screen to Dr. Sadanand for his deliberation. Dr. Sadanand, please.
Ma'am, just one announcement. You may post your question in a Q&A section. Maxime, yeah. Yes, Dr. Sadanand, please.
Yeah, thank you, Shubhangi, for such a nice introduction. First, good evening, everybody. And I'd like to thank Shubhangi and the Department of Microbiology and Nagpur Veterinary College for inviting me for this webinar. Is there any problem?
No,
Okay.
continue, please.
Yeah, so thank you for inviting me. So now I'm going to share my screen, right? Okay, share. Can you guys see my screen?
Yes, yes, everything is good. Yeah.
Okay, so before starting my actual talk, I'd like to introduce my lab and my lab members and my area of research. So, and before that, I'd like to disclose my fundings. So I have a couple of projects funded by NIH. Right now, recently, one of my grants just got funded by NIH on Arginase 1 and muscle and bone. And then there is another Q1 project I'm working on. That is on the HDF1 and the microRNA and related to this stem cell. And there are like other projects with my collaborator. And recently we just finished a couple of projects. So this is the building where I work. I have two labs. One is on second floor and another on third floor. Basically on the second floor, we do all the cell culture and handling of human tissue. And on the third floor, we do most of the biomechanical stuff. And this is my lab members. I have a couple of postdocs and some students that are working with me. They almost all they are very highly talented and very hardworking. So basically, COVID-19 is not my research area. I'm working on stem cell aging and the sports medicine, and I did some stuff on the spine area also. And recently, in the last 5 years, I did some work on aging research, and we published a number of manuscripts in this area. In stem cell research area, we did a lot of work basically on how the stem cell differentiating to the bone cells. So these are the recent publications. And then in the area of sports medicine, we did some on the patellofemoral pain and joint tissue, and some on the growth factors. And I did some stuff on the osteoarthritis, on basically on the human tissue samples. So, and some on the spine. Why I'm showing this? If there is always an possibility of an collaborative work, because some of you guys might be in the Department of Medicine, surgery. So if you see these things and if you like, and if you guys want to do some kind of any interactive research, then definitely I can help you guys, or even we can submit some Indo-US projects, and that may be beneficial for you as well as me. So coming back to the COVID-19 related research, so as I told you, COVID-19 is not my area of research, but this is the natural disaster come to our way, and I think we need to respond in a really nice way. And I felt like this is my obligation, that I know something about the microRNA, I know the virus really well, so why not do something else in this area? So, and another important thing, I can see there are a lot of people, they are listening to me. Some of them may be the student, some of maybe the faculty, and some of maybe the personal working in the field. So it's very difficult for me to make a presentation that everybody will understand. So I just try to keep everything very simple, and I'm not going into the molecular biology. so that you will sleep on your chair. Okay, so this talk I'm dividing the two parts. One is on MicroRNA and COVID-19, which is really interesting. But there is another project I'm working on, what I'm seeing COVID-19. That is also interesting. So first, let's talk about the MicroRNA and COVID-19 project. So first, this is the basic thing we need to understand, that what is the cell and how it works. So on my screen, you can see this is the nerve cell, this is the myoblast, and this is the fat cell. And if you ask a question yourself, is DNA sequence in all cells are identical? And I think most of you guys might answer yes. Right? And the answer is yes, that's right. But then there is another question, why each cell have cells behave different, or why myoblast different than the fat cell or nerve cell? So this is a valid question, and I think most of you guys might answer, or maybe get confused why nerve cell look like a nerve cell and why fat cell look like a fat cell. Basically, DNA is the same, genes are the same, but the gene expression is different. But again, the genes are the same, right? So why there is a different looking cells? So most of the things are because of epigenetic regulation. And I think tomorrow, Dr. Bass is going to give a presentation of epigenetics. He's a fantastic guy, and he can tell you the other portion of methylation and histone modification. So there are typically three broadly epigenetic regulations. One is the histone modification, methylation, and third is the microRNA. And these are the main component play important role in the gene expressions. So what happened? Every cell's expression, suppose for example TNF alpha, but in some there will be less TNF alpha expression because the microRNA is inhibiting the expression of that particular gene, and that's how it's regulated. So if we know how the microRNA and epigenetic thing regulate, we can design our research depending on what your goal is. So first, let's talk about the microRNA. I'm just trying to tell you basic stuff first. So we know the RNA. RNA is a ribonucleic acid, and it can be divided into basically two parts. One is a coding RNA, second is non-coding RNA. So when we talk about the coding RNA, there is an mRNA, messenger RNA, and then there is a non-coding RNA that is called as transcriptional RNAs. So this transcriptional RNA help in transcription of genes, and basically those are the ribosomal RNA and the tRNA. And there are the another kind of an RNA called as small RNA. And not only means we can there is in small RNA there is a large non-coding and all those things. I'm not going into detail, but let's talk about the small RNAs. So there are basically three kinds of small RNAs. One is the siRNA, second is microRNA, and third is piRNA. We'll not talk about siRNA and piRNA because that is a separate topic. We'll just talk about the microRNA. So basically, microRNA, they are the guardians and fight against the unwanted genetic material. For example, if there is a virus entering the body or in any cell, and if the microRNA see any complementary sequence on those viruses, it goes and bind and cleave and destroy it. Or in a normal way, what happens? There is an overexpression of a particular gene that we don't want, and if there is a microRNA complement to that, it will destroy that microRNA or it inhibit the translation of that particular mRNA. So that is the basically microRNA. So I'm going a little deeper. So basically, this is a structure of an microRNA. Okay, most of the microRNA in similar, only the change in the sequence of an microRNA. So this is the cell, and this is the nucleus, and this is cytoplasm. Basically, what happened when the gene transcript? So microRNA also have a gene. Means that is the means don't think that the normal gene and the microRNA are different. They are the same thing, only there is a difference that microRNA doesn't code for any protein, but normal mRNA code for a protein. So I will just break it down in a very simple way. So basically, this is the pre-microRNA. This is the mature microRNA. So when there is a gene transcript, and this is, you can say, the pre-mRNA. So yeah, this one. And there is an enzyme called as DORSA. It cleaved here and make an like this. This is called as this is pre-mRNA. And with the help of exporting protein, it comes out, and again it cleaved with the dicer, and it became the double-stranded the microRNA. And this is the 5p, and this is 3. So there are like when it form a mature microRNA, there are two kind of microRNA, because and then this gets separated. So here you can say is a pre-microRNA, it comes out to exporting 5, and then there's a dicer, it goes get cleaved, and then there is a double thing. So here what happens, this microRNA will bind to the mRNA if it sees this complementary. Okay, so for example, here you can see this is the mRNA and this is the microRNA. So when A and C, G and C is there, it's the complementary, and it form an hydrogen bond, and then it cleaved, or it will suppress the or inhibit the expression of that protein. So that's how this is the basic mechanism how the microRNA works. So coming back to the COVID-19, and so when the lockdown started, so I was thinking what we can do to play important role in this COVID-19 research. So we come with an idea, why not look for the microRNA which is targeting COVID-19 and connect the dots why there are some people getting more infected, some people not infected, some people are dying, some people are not dying, and can we correlate with that? So that's how we come with this idea, and we wrote a manuscript. So this is on my screen. You can see this is an infamous picture. You can say this is the COVID-19 virus, and this is the spike protein, and this is the RNA. Okay, so let's understand how this virus attack the cells and what usually do. So this is the cell, and you might have heard in news and everywhere that is a angiotensin converting enzyme 2. So basically, this is the receptor. So this virus bind to this receptor because there is an this virus has a strong affinity for this receptor. So once it is get bind here and get inside the cell, once it's inside the cell, genome is there and it get replicated, and then it get translated, get assembled here and come out from the cell, and that's how it replicates. So now we know how this replicate, and we know there is a genome here. So we can target somewhere here using a microRNA. Okay. So now we know that this virus is there and infecting this many people, and there is a lot of mortality going on. So there is only one way out, that is a development of a vaccine. But development of vaccine is not an 1-year project. It takes a long time to develop a vaccine. Okay, so the targeted therapeutics is an important contribution, and a lot of people, they are working in this area to combat this virus. So there are two types of an targeted therapeutics. One is like a neutralizing antibody. So we can develop an antibody against this the spike protein, and we can neutralize the viral replication. So for example, you might have heard about the plasma therapy. A person who get infected, we can collect the plasma from that person, which contain the antibody against that particular virus, and then we can transmit to the patient. And that's how the patient can fight against the the COVID-19 infection. Or there is the another way we can stop this viral replication by using antiviral drugs. And recently, you might have heard about the remdesivir. So, and this is a life-saving drug. The only problem with these days is remdesivir, the people giving when the COVID-19 patient is very late stage, like I don't think they have enough stock. I don't know what's going on, but this is a really nice drug. And how it works? So when the virus get enter in the cell, this is an RNA virus. So you have to understand. So when it's an RNA virus, and this virus need an RNA-dependent RNA polymerase enzyme. So remdesivir, it bind to this RNA-dependent RNA polymerase, and here inhibit the replication, and that's how it's working. Okay. So coming back to the microRNA, as I told you, the microRNA, these are the small length nucleotides around 22, have an ability to inhibit the viral replication. So, and recently there are a number of manuscript people they have published already, showing that it work in a lot of other diseases. So what we did, we collected the download the sequences from the NCBI, and we compared. And you might have heard about the SARS with an COVID-19. In 2002, 2003, 2004, there was an outbreak of an SARS, which is also coronavirus. And that virus was more dangerous than this virus. And so initially I asked a question, why this virus is different than those that SARS and the Mediterranean virus. And I compare the genome sequence, and we found that there is a 78% homology between the SARS, which outbreak in 2002, 2003, and this COVID-19. So there is a 20% difference between that virus and this virus, and that might be the problem. But another important thing, this is very weak virus. This is weak and not that dangerous compared to the SARS. The mortality rate of the SARS virus was very high. And this virus is very weak, and that's why it's making him stronger. Now you ask me why you are saying this is a weak virus and causing this many problems. Because what happened when the infection takes, it takes 5 to 6 days to get the symptoms. So once you have our infection, you are going out in the market and giving everybody else. Just imagine if it is a very dangerous virus, the mortality rate is high, cytopathic effect is very high. What will happen? Once you have infection within 24 hours, you will have an high fever, and you start showing the symptoms, will not go out, right? So that's why the infectivity is more, but the mortality rate is less. But there is a lot of people are getting infected, so relatively people are dying more. So don't take me wrong what I'm saying. So at the nucleotide level, you can see there is not much difference between the viruses. Like, and we analysis the virus from the Canada, China, Germany, even in India. So all these strains of COVID-19, they are very similar, and there is not much change. And you might have heard a lot of rumors, oh, this is going to be very pathogenic virus because there is a lot of mutation. No, that's wrong. As per my knowledge, there is some mutation because this is RNA virus. Definitely there will be mutation, but it's not changing its pathogenicity. That's what I noticed. So then we try to identify what are the microRNA targeting the genome of this virus, and we found that around 850 microRNA targeting the SARS virus, which was outbreak during the 2003-2004, and around 900 microRNA targeting the this COVID-19. And then we compare what are the microRNA which are common and the different. So we found that the 588 microRNA that are common for the SARS as well as the COVID-19. So imagine if somebody might have worked during 2003 and identify maybe 5 or 6 microRNA targeting the coronavirus, we might have used those microRNA for recent outbreak. So it's very important for a future also. I don't think you guys may ask, oh, that's okay, it's fine, this microRNA targeting the COVID-19, but can we use this microRNA for this recent outbreak? No, never. I think, but imagine this coronavirus is not going anywhere. There may be some outbreak in 2025, maybe 2030 or 2035, maybe after 5 years, maybe 10 years. But we know that these coronaviruses are very concerned. So even if our 5 or 6 microRNA which are targeting, I bet you because of 80% of homology, this microRNA will be very useful in the future. So that's why, and we are aggressively working on doing some lab work also. Okay, so we found around 900 microRNA targeting this COVID-19. Out of those, like this is the target score. Target score means the probability of this microRNA targeting COVID-19 is 99%. Means when we do the analysis, there is always possibility that microRNA might target, may not target. So, but it depends on the homology, it depends on the hydrogen bonding, it depends on thermostability. So those are the factors play important role when we try to identify the microRNA. So these are the selected microRNA right now I'm working on, and this microRNA, for example, 15B5P and 15A5P, this microRNA, the sequence is different, but seed sequence is the same, and they target 16 sites on the coronavirus. So maybe even if 10 sites are targeting, then that's more than enough. So we are working on this microRNA extensively. And then what we did, we tried to identify what actually happened with an aging. We know that aging, diabetes, cardiovascular, all these are the factor which affects the COVID-19 severity and mortality. So, and this is known fact that with an aging, we lose a lot of microRNA. A lot of microRNA goes down with an age. And that might be another factor causing this virus replication, because we don't have that microRNA in your cell, or if you have that microRNA in your cell, but the level are less and the virus is taking over. And these are the microRNA. These are goes down with an coronary heart disease, kidney disease, cancer, aging. So you name it. These are the microRNAs which are downregulated in these underlying conditions, and these are the underlying conditions which is known for an COVID-19 infection. So this is a really interesting study published in I think around 2013. And I think most of the people that are listening me are veterinarians. So I thought this is a really a nice example. So this is a porcine reproductive and respiratory syndrome virus. This study published in, again, I said, I think 2013. So this 181 microRNA target this virus. And they did bioinformatic analysis. They found that this microRNA target, and they published this one. So you can see, if you guys are the microbiologists, you know what is the cytopathic effect. So they culture the cells, and then they infect with an that virus, and then also transfect with an microRNA mimic. And you can see this is the control, and this is a microRNA mimic. Wherever they put the microRNA mimic, the cytopathic effect is less, and there is a more cells compared to here. There is more cell death, you can see. And this is the control, and you can see here the microRNA, and you can see the title is going down, and here also same thing. So it looked like 181 microRNA prevent uh, viral replication, right? So they did really one good experiment on the pigs, and they use a nasal as nasal therapy. So they use 181 microRNA mimic through a nasal, and this virus is basically go through the uh, means infect through the nasal, and that's why we use the mask. So if we develop some of the microRNA and give to the nasal string, it might prevent the viral replication, and we can combat this disease. So this study, what they did, you can just concentrate first on this figure. So they took a pig and infect with a virus, and they gave an first dose of a microRNA mimic here, and second dose here, right? And you can see all the animal died at 16, at day 16 of an control. But you can see here, the pig which got the microRNA have prolonged life. But they, in human condition, will not just give one thing and then let them die. We'll give like maybe 3 or 4 injections, and other thing we give other life support. So it look like we can really combat this virus with an microRNA therapy. And here you can see the temperature of an animals are down here, and this is the survivability. So my point is that 181 microRNA can stop or delayed, or we can say improve this survivability of the pigs, and we can use same kind of therapy for human in an outbreaks. Okay, so this is about the microRNA. I hope I try to explain very easy manner. I try not to use very scientific words or whatever. So let's go to the second project, and this is very close to my heart because I spend a lot of time working on vitamin C through my career. And this is very interesting. Why I'm saying that? Because these days, WhatsApp, Facebook, and all this social media making a lot of buzz about small thing, and we forward each other without reading or without taking responsibility. So I request you guys, when you get any message, just don't forward. First go in the depth and listen or read, and first do the investigation and then forward. I got a lot of messages saying, oh, vitamin C help and do this and that. And these days people, they are doing the kara and all those things. Ramdev Baba making his own medicine. So all these are things going which divert the actual research. So I thought, why not? I have some knowledge about what I'm seeing. And so we wrote an article, and it is under review in the journal Nutrition. It's under, we got the comments back. It's a minor revision. I'll submit maybe on Monday or Tuesday, and I hope within a month we'll have that manuscript online. So let's talk about vitamin C. Everybody knows vitamin C. Even my mother knows vitamin C, and she said, "Okay, why don't you eat, drink, and your bones will get strong." Yeah, that's good. But when you go to doctor, they recommend vitamin C for your bone health. No, they recommend vitamin D. Anyway, so vitamin C is a water-soluble vitamin, and it's very important vitamin because our body don't make vitamin C. And a lot of research you see on the mice make their own vitamin C. During the evolution, there is a mutation on one enzyme called GULO. That's an enzyme which play important role in vitamin C synthesis. But what exactly happened during evolution? There is a mutation, and we cannot make our own vitamin C. We need an vitamin C from outside. But again, vitamin C is water soluble. Once you drink, it can absorb in your intestine, but it will not go inside the cell because the water soluble. And our cell is an lipid bilayer membrane, which hate the water. So to get into the cell, we need a transporter. And we got a transporter called vitamin C transporter. So suppose this is the cell and this is the lipid bilayer membrane, there is a vitamin C transporter. It doesn't matter how much vitamin C you are taking. If you don't have vitamin C transporter, it will not go inside the cell. That's period. Okay. So this is very important. So we published a paper in 2020 in stem cell research showing that my lab is the first one to demonstrate that vitamin C transporter present in the stem cells. So we published this paper in 2013, and in this paper we have shown that with an age, the vitamin C transporter goes down. These are the mice. And again, I said vitamin C don't make what mice make their vitamin C. We don't make our vitamin C. Okay, but this transporter play important role. So in 12-month mice, you can see there is a good amount of expression of an vitamin C, but it goes down 50% with an age. That's good thing. No, that's bad. Okay, but and how can, so we try to identify what are the factors can upregulate this transporter, and then we can give vitamin C so that, or we can come with a drink which contain and something which will increase the vitamin C transporter expression, and then you can have some vitamin C and your body will get vitamin C. So we thought like, let's try vitamin C. We treat the stem cells with vitamin C, and we didn't see any change in vitamin C's uptake. So uptake will increase only if the vitamin C is present. But when we treat with an dexamethasone, the vitamin C uptake goes up within 24 hours, 48 hours, and in 4 days. So this figure shows that the dexamethasone upregulate the expression of vitamin C transporter, and it helps in taking the vitamin C inside the cell. And in last 1 month, you might have heard a lot about the dexamethasone or steroid hormones that when you give to the COVID-19 patients, severe patients, they are recovering and the mortality rate is going down. So at this stage, whenever you go to the hospital, if there is somebody severe, they are trying to two drugs. One is remdesivir, another is an steroid hormone, maybe dexamethasone or some other steroid, but steroid helps. So what my hypothesis is, and this is partially, it's not like, and when you hypothesize, is that 100% correct, okay? So what I'm thinking, maybe when the patient getting the dexamethasone, the vitamin C transporter goes up, and they're getting a vitamin C inside the cell, and they are boosting up the immune response. And so that's what we argued in our master. And this is the another article they published saying that a cheap steroid is the first drug shown to reduce the death in COVID-19. So this is really a good sign. And maybe if the my work come in there, and we can say that the dexamethasone effect is partially through this transporter. Okay, so another important thing, vitamin C disparity in the gender. Okay, I don't know how many people you guys know that this COVID-19 is disproportionately affecting the gender. The female basically are less affected by this COVID-19, and you will be surprised to know that the vitamin C levels are higher in female compared to the males. And in COVID-19 patient, the female having less, this is the female, and there is a less death compared to the males. You can see here, there is 13%, 31%, 45%, 51% people, they are dying with an older age, and the females, they are not dying. Yes, they are dying, but it's not like in the way the males are dying. And I'm arguing that maybe because vitamin C levels are more in the serum of in females, that might have partially protecting this, and that's why there is an less severity. So you can see there is another article. They are shown that the male are more serious condition than the females. Uh, okay, and coming back to the African American population, again, this vitamin C transporter is very funny. It has a polymorphism in a lot of African American population. So because of the polymorphism, it doesn't matter how much vitamin C you are taking, the vitamin C is not going inside the cell after a threshold level. So that when you take in vitamin C, if there is a polymorphism in this transporter in your system, then there is a level can go, but it doesn't mean like you are taking a lot of vitamin C and it will go inside now. And other thing, in African American population, because of their social economical status is little less, they tend to eat a lot of junk food, or they don't get enough food or whatever, that put a lot of stress on the system, and the transporter is down regulated. And in underlying conditions, in diabetic patients and the cardiovascular complications, it is well known that the vitamin C level is down. So again, in underlying condition, vitamin C level is down, and the severity and the mortality is more. So coming back to the oh, this is another interesting interesting study. This is an double-blinded placebo-controlled study. They gave an intravenous vitamin C to the severe acute respiratory failure patients, and this is the thing also happened in the COVID-19 patients. So you might have seen that a lot of articles, or even in popular articles, saying that vitamin C really helps, but those are not the double-blinded placebo-controlled studies. So double-blinded placebo-controlled studies means even doctor don't know what they're giving, and even patient know what they are getting. So that's why the Moderna is doing the double-blinded placebo vaccine trial, which will tell us whether this vaccine is effective or not. If you know that you are giving a vitamin C to your patient and it gets better, you report, and if you don't, then you blame somebody else. So, but if you want a really valuable data, you have to do the double-blinded placebo-controlled study. And this is the large study, um, and they have shown that vitamin C compared to placebo did not significantly improve the organ failure. And most of these patients are aged, and maybe the vitamin C transporter level is down. And if you are giving through intravenous or whatever, it's not going inside the cell. And one important thing, what happens when you take a lot of vitamin C and you have less transporter? Vitamin C accumulates in an extracellular space. So vitamin C is an antioxidant, but sometimes if it's accumulated too much, and it became oxidant. So instead of getting a better result, that may have an opposite effect. So that's very important thing. Okay, so this is an interesting table, and just concentrate on this my question. So male and female here, and let's see vitamin C levels. Vitamin C levels are less in male, and they are more severely affected by the COVID-19, and mortality rate is high. In female, the vitamin C levels are more, and COVID-19 is less. Same thing, Caucasian white versus African American. You can see there is in Caucasian population, there is more of vitamin C level and less severely affected. African American, low vitamin C level and highly affected. Same thing with the underlying condition like diabetes, hypertension, COPD. You can see there is a less open vitamin C, and there is a high prevalence of an COVID-19 infection, and the mortality rate. When there is no complications, there is a less often chances of an severely affected. And same with an age. the young age people, they have a high level of vitamin C, and they are less affected by the COVID-19. Once you age, it goes down, and the rate of an infection goes up. So overall, this is interesting area of research. And right now, WHO are conducting 30 clinical trials based on vitamin C. So I thought this is my responsibility to write something, because when doctors or people they do the clinical trial, they cannot think about, because they have expertise in a limited thing. Suppose if I give my input, that might be correct or wrong. It doesn't matter, but this is my hypothesis. So there are like 30 clinical trials going on. Some people they are giving high dose of vitamin C, that's in China, and some people they are giving vitamin C plus hydroxychloroquine. So there are different combination of a drug they are giving, and we don't know what will be the outcome. But if we analyze the data, depending on our knowledge, if we publish this paper, and somebody from this clinical trial says, oh, vitamin C transporter play important role. So they might analyze the vitamin C transporter in a, in a macrophage or whatever the sample they have, and they can correlate, oh, this person died and this person have this kind of an transporter is down, or this person survived with an vitamin C therapy because the vitamin C transporter is up. So that's what my perspective, and that's why I wrote this manuscript, and I hope it will get accepted within some days. So coming to the conclusion, what I feel like vitamin C is really good to boosting the immune response, but in a young patient or young population, but with an age, I think, and race, so it's gonna affect. So overall, I feel like vitamin C is good, but it also depends on the various factors like age, race, transporter, and polymorphism. Um, so again, this is my lab and collaborator. So these are my students and my postdoc, and I'd like to thank my all the collaborator and the funding agencies for helping me out during the last 10 years. And I like to thank my beautiful wife and my son. They contribute really well in my research. Thank you very much.
Thanks a lot, Dr. Sadanand, for your presentation. Almost 11 questions that I have seen in front of me. So we will take one by one.
Sure.
First of all, Tulendra Singh Dikshit, he want to ask, can SARS vaccination provide cross protection against COVID-19?
Yeah, whatever I read till now, no. The answer is no, because the spike protein is a little bit different, and the immune response developed in the SARS is different than the COVID-19. So there is, means I haven't heard anything in that area. So I don't think it's gonna work. I can't hear you.
Next question from Mamta Pandey. The question is, why do you think of higher level of vitamin C in females as compared to males?
Yeah, that's a good question. I don't have answer for that. I don't know. Maybe the transcriptional regulation of that particular gene is different in male and female, because maybe there is a reproductive hormone might be playing a role. Maybe estrogen is regulating this transporter, and that's why there is a more expression of an that gene in the females.
The next question from Dr. Prabhakar Tembune. The question is, single gene can be targeted by multiple miRNAs. In case of COVID-19 coronavirus spike genes, how many miRNA did you find? Can it be used for therapeutic miRNA?
Oh, thank you, Dr. Prabhakar. Nice to hear from you. Okay, so the genome of this coronavirus is around 29 KB. And we just screen for all, and you don't have to design specifically for a spike protein. What I understand, if you somehow even target RNA dependent RNA polymerase or any kind of protein which are important for the assembly of a virus, you can inhibit the replication. And at this stage, I cannot tell you how many I found it, because it's not on my top of head, but I can text you. Okay.
Okay, thank you. Now, next question from Sujit Kolangar. Is there a difference in miRNA pool considering the racial diversity of humans?
Yeah. The expression level is maybe different, like, because uh yeah, I think race, sex, and the fooding, the habit, all they play important role in the expression of an microRNAs. And I feel yeah, I think it's going to be really interesting. So that's why we started really a nice project in which we collected the nasal sample from the various race, like an African American population and white Caucasian, male and female, and we are analyzing how these the COVID-19 affecting the expression of this microRNA, and that's going on. And another important project I just like to tell you guys, we're working on, like in some people they get very severely affected, some people not, and we thought maybe there is some nasal microbiota playing a role. So we collected the nasal sample, and then we isolated the DNA, and we sent for the sequencing for the bacterial population. And it's going to be really a cool project. Thank you for asking a nice question.
Next question from same, that is Sujit. Can miRNA be a therapy of future for respiratory infections?
Yeah, I think, it's a really nice question. And I feel, yeah, not now, but maybe in the coming 10 years, maybe 20 years, definitely. You can say that till now there is no mRNA-based vaccine, but Moderna is making that one, and in the market in November, December sometime. So till now, there was not a single vaccine based on the mRNA. Most of them vaccines are the DNA vaccine, or maybe attenuated or killed vaccine, all those kind of vaccines. But the science is developing so much that we have to think in a new way, new therapeutics. And I think you are right. Like in Moderna making an mRNA vaccine, which is not used yet, but it will be in the market in November, December. So same thing, it will be true for the microRNA in the future.
Ma'am, unmute kara.
Yeah. Next question from Rukshana Shah. How do you look at the reinfection in COVID cured patient?
Okay, this is very controversial, and I don't want to comment on something I don't know, but I can put my point there. So I heard that there are some reinfection in the South Korea, but that may be because of a dead virus present in the body, and the PCR-based diagnostic method can detect the dead virus. So that's how it look like, and there is an reinfection, but it's just a dead virus. So that's one thing. And suppose if somebody get an infection and they were not able to produce an enough immune response, then they might get it maybe after 6 months if there is no memory cells, or I don't know. I feel like if somebody get a really infection and they have a strong immune response, they will not get infected again.
Okay, next question from Shaina Sayed. When so much about COVID-19 pathogenesis is already known, why is developing vaccine taking so much time?
Okay, I think that's an I think according to you, it's taking so much time, but this is the record-breaking time we are getting the vaccine. Vaccine development is not an 1 year work. It's 10 years or maybe 15 years because we have to go to phase 1, phase 2, and phase 3 clinical trials, and which take long time. And for the COVID-19, I think this is the record-breaking fastest vaccine developed in whole universe. No one has done like this before, and I think if this Moderna and the another Oxford vaccine come out in November, December, that will be faster. But you have, like if somebody give you vaccine and if it's not effective and or maybe have side effects, then you will blame the company. So you have to keep balance between what you are getting, right? Yeah, go ahead.
Okay. Next question from Ravikant Khobragade. Kindly elaborate about the pathogenesis of COVID-19 in felines.
Oh, so I heard that there are some of the cats got infected. I don't know exactly the reason, but I think there is an AC2 receptor maybe similar like in humans, and that's why they are infected. Um, and yeah, that's what I can say that. But I don't know much about the pathogenicity.
Okay. Next question from Jeevan Keshekar. Neutralizing antibodies versus miRNA, what has promising therapeutic value?
I think at this stage, I think the antibody based therapy is the option. The microRNA, I think that's what I told you. It will take in maybe 5 years, maybe 10 years, but this is the future thing. This is not for this outbreak. It's not that I'm going to make any therapy for in coming days. It takes a long time and a lot of efforts from different researchers. And yeah, so I think the plasma therapy and the vaccine and all these vaccines will definitely help getting back to the track.
Okay, last question we take, sir. Can virus delivery vehicles be effective for delivery of miRNA as a therapeutic?
Yeah, so the delivery method of an microRNA is because they're like in other words, I can say that these are the different I don't know how to put in words, but the delivery method, there are different delivery methods. Some are delivery method just to deliver, and some have an, for example, for a vaccine, you deliver a vaccine with an adjuvant and you boost up the immune response. For the microRNA, you are not boosting the response, but you are just delivering, and there are like a lot of different methods, and each have a different advantage and disadvantage. So at this stage, I cannot say which is better, which is not good. I think uh, at this stage, I don't have an answer for that. What method we'll use, but definitely, I think if for a respiratory, I like to go for a nasal, some like a spray containing something which stress the cells and microRNA go inside the cytoplasm.
Okay, thank you. Now we come for the concluding remark. So, good evening to all. I, Dr. Shubhangi Varke, Organizing Secretary, feel great honor to propose the vote of thanks to all who have helped us for making this international webinar such a resounding success. I would like to express my heartiest thanks to Colonel Professor A.M. Paturkar, Honorable Vice Chancellor, Maharashtra Animal and Fishery Sciences University, Nagpur, for his valuable guidance, constant encouragement, motivation, and advice for organization of this international webinar. Sir is a true inspiration to all of us. We are really grateful to him. A special thanks to our distinguished today's speaker and eminent scientist, Dr. Sadanand Fuljale, Assistant Professor, Department of Medicine, Center for Health Aging, Cell Biology and Anatomy, Medical College of Georgia, Augusta University, USA, for accepting our invitation in such a short notice. On behalf of the organizing committee, I take this opportunity to wholeheartedly thank and congratulate him for his expert deliberation. The way he explained the topic is outstanding. Today, you have inspired many young minds to take up an interest in research related to microRNA. Beside critical situation in the USA due to COVID-19 and his busy schedule, the amount of risk and effort he has put in to make this event successful is surely commendable. Our best wishes are always with you for all your future endeavors. I must say that we all are inspired by your enormous research work. Thank you once again. I am thankful to Dr. S.V. Kavitkar, Associate Dean, Nagpur Veterinary College, Nagpur, who has shown keen interest in organization of this event, also for permitting and supervising us with vision and leadership. His constant encouragement always supports us. I express my sincere thanks to University Librarian Shri Sunil Gavandeji for providing all the necessary facilities for smooth conduct of this webinar. The word falls short to our knowledge partner, Dr. Vijay Nilkant, Product Manager at Vetina Healthcare, Pune, and his team for their tireless contribution they have made. I admit that the webinar could not have been possible in such a short span without their help. I personally feel that behind every successful event, planning and team of committed faculty is required. We have fortunate enough to have a motivated team and dedicated colleagues of our department. I acknowledge them for their work and efforts. Last but not the least, I am thankful to all the participants to attend this webinar from different educational institute and stream. Thank you for your overwhelming response. I'm sure this webinar will prove very informative to all of you. I'm thankful to one and all for being a part of this knowledge sharing feast. Due to overwhelming response, time may require to deliver participation certificates, so please bear with us. Tomorrow, same time, there will be another international webinar on epigenetics therapy on cancer by Dr. Manish Bess, Assistant Professor from USA. I request all the participants, please be live with us and attend the webinar. Thank you for sparing your valuable time. Looking forward for more such scientific deliberation. Have a great evening. In the last, with Indian tradition, I must say Namaste. Thank you.
Yeah. Ma'am, we have with us our CEO, Prakash Khere. He wants
Yeah.
to share some words. Sir?
Yeah.
Hello.
Hello, sir.
Hi.
How are
Good
you?
evening, sir.
Yeah, good evening. So I wish to thank you, Dr. Sadanand, and you for organizing such a wonderful webinar. Vetina has been very active in disseminating the knowledge, of course, in the veterinary field for the benefit of farmers and the vet professions. So we are conducting regularly the webinars on different topics, and I'm sure this would benefit to all of you. So I wish on behalf of Vetina, thank you for all the participants and look forward to be associating with such a scientific forum. Thank you very much.
Thank you, sir.
Yeah, and I like to thank for you guys for doing such a nice job. And I think what I feel like in Nagpur Veterinary College, and you guys should organize such kind of thing, regularly, so that the students should get inspired. Here, like there are a lot of Indian students studying here are faculty members, and I think if you contact them, they can deliver maybe one lecture or two lectures every month, and that definitely will help our students, inspire them. Because I know that I grew up in Nagpur Veterinary College, and what I am today is just because of that environment. But it depends on also what kind of environment you are getting, and if we collectively do something like this, and I'm so proud of Shubhangi and whole department and Dr. Prabhakar that they're doing really tough. And thanks for inviting me. And if you need anything, if you want to collaborate with me on any kind of projects, I'm always there for you guys. And just contact me, and you'll get my email ID from anybody else, and or even you can Google it, you can get it, and you can contact me. I'm always there for you guys. Okay, I love you guys. Thank you very much.
Thank you, sir. Thank you very much, and thank you, ma'am.
Thank you, sir, for your kind words, Dr. Sadanand Fuljale. And good evening to all of you, and good morning to USA. Thanks. Can we leave?
Yeah.